In 1982, researchers counting cells in the basal forebrain made an observation that would define Alzheimer's research for half a century. They found a profound selective neuronal loss in a structure called the nucleus basalis of Meinert. These CH4 neurons broadcast acetylcholine across the cortical mantle, but as the disease progresses, these vital supply lines wither and collapse. This physical destruction established a mechanistic assumption. The field concluded that possessing a cholinergic phenotype is the inherent risk factor that destines a cell for destruction in Alzheimer's disease. That specific biological assumption directed 4 decades of pharmacology, yielding an entire class of drugs designed exclusively to replace the acetylcholine lost in the forebrain. The foundational hypothesis mapped the cortical broadcast in granular detail, but it entirely ignored the brain's central switchboard. Between 1983 and 1989, comprehensive neuroanatomical mapping revealed that the human brain actually contains 6 cholinergic sectors, not 4. This 3D model isolates CH5 and CH6 in the mesopontine tegmentum. Vector lines illustrate they provide the sole major cholinergic innervation to the human thalamus. Microscopically, they match the doomed CH4 neurons perfectly. They are large, cholinergic, and accumulate dense Alzheimer's tau protein. Yet modern quantitative analysis proves a stark biological reality. These neurons carry every recognized risk factor. They bear the pathology of the disease, and they do not die. The survival of CH5 and CH6 definitively refutes the bedrock premise of the field. Being a cholinergic neuron is not a sufficient condition for vulnerability. For 30 years, the scientific literature contained a glaring discrepancy. In 1988, a highly cited morphometric study reported a 30% loss of these pedunculopontine neurons in Alzheimer's patients. This split-screen schematic explains the contradiction. On the right, we see the modern approach from a 2018 multidimensional quantitative analysis. Using design-based stereology, researchers found that the total neuronal numbers in the disease cohorts were statistically identical to healthy controls. Looking at the older method on the left, we can identify the specific artifact that skewed the historical record. The 1988 study applied a strict size criterion, deliberately counting only the large neurons. When a cell accumulates neurofibrillary tangles, its physical response is somatic atrophy. The cell shrinks. In the morphometric grid, it drops below the size threshold and is excluded from the count. In the stereological volume, the shrunken, tangle-laden cell remains physically present and accounted for. This cross-section shows that the survival of these cells is not a generic feature of brainstem resilience. Mere millimeters away, the locus ceruleus is routinely annihilated early in Alzheimer's progression. In the exact same tissue samples examined in 2018, the disease also severely depleted the GABA-producing gigantocellular nucleus. Furthermore, the pedunculopontine nucleus is entirely capable of dying. In progressive supranuclear palsy and Parkinson's disease, it suffers up to a 60% loss of its cholinergic population. Alzheimer's pathology reaches the mesopontine tegmentum and aggressively destroys neighboring tissue. The survival of this specific cholinergic population is an anomaly that requires a precise molecular explanation. If transmitter class, cell size, and location cannot explain this selective vulnerability, what exactly separates the doomed forebrain cell from the surviving brainstem cell? This cellular schematic illustrates a cytochemical profiling study published in 1989. On the left, the vulnerable CH4 neuron carries high levels of nerve growth factor receptor proteins on its membrane, specifically TRKA and P75. On the right, the CH5 neuron completely lacks them. Those receptors bind the forebrain neuron into a strict trophic contract. The cell cannot manufacture its own survival signal. It is entirely dependent on a constant retrograde supply of nerve growth factor transported backward from its cortical synapses. As the disease dismantles the cortical target, the synapse breaks down. The retrograde transport apparatus fails, severing the supply line and starving the parent cell of its required survival signal. Because the mesopontine neurons of CH5 and CH6 never possessed those receptors, their survival is uncoupled from the degradation of the cortex. Alzheimer's disease does not hunt cholinergic cells, it kills trophically dependent cells. The pedunculopontine nucleus survives because it never signed the contract. This leaves us with a specific biological state, a standing relay. This is a nucleus that retains its structural neurons, but has its output suppressed by the tau accumulating inside them. Because the pedunculopontine nucleus is the primary generator of REM sleep, sleep architecture provides the most sensitive, continuous readout of the structure's functional state. This graph charts polysomnography metrics and ENS disease progression. In the Framingham cohort, sleep data proved that lower REM percentage and longer REM latency strongly predict incident dementia years before diagnosis. This pattern perfectly matches the mechanics of a standing relay. An underdriven but living generator takes longer to assemble the required firing threshold, resulting in delayed REM, and it lacks the power to sustain it, resulting in shorter REM. A dead nucleus would simply result in zero REM. Crucially, this impairment does not cause REM sleep behavior disorder. That condition requires a lesion in the separate sublateral dorsal atonia circuit, a circuit targeted by the synuclein proteins of Parkinson's, not the tau proteins of Alzheimer's. The early sleep disruptions preceding Alzheimer's dementia are the precise, measurable output of a struggling, intact mesopontine nucleus. The dominant model of Alzheimer's disease is subtractive. Researchers and clinicians assume that functional loss occurs in direct proportion to cell death. This creates a rigid diagnostic trap. Because the pedunculopontine neurons shrink rather than die, standard volumetric neuroimaging, like structural MRI, will fail to detect the dysfunction entirely. You cannot image missing volume if the cells are still standing. Organizing therapeutic logic solely around preventing cell loss creates a blind spot for neural nodes that remain structurally preserved but functionally dormant. To prove this functional perspective, researchers must execute a specific, unperformed experiment. They must measure vesicular acetylcholine transporter immunoreactivity in the thalamic relay nuclei to verify if the channel is still transmitting. If the channel is compromised but the source cells remain, the implications are immediate. A fully populated nucleus with intact neurons can theoretically be driven from the outside, either pharmacologically or via targeted deep brain stimulation. A nucleus that has lost 3/4 of its cells is an untreatable ruin. A nucleus that retains its cells is a rare, vital target waiting to be reactivated.